So what I thought I would do this morning is look at four key questions, or try and cover four key questions for you. First of all, what is the genetic architecture of the types of complex traits that we deal with? So by complex traits, this is actually a phrase from human genetics that human geneticists use really widely. But in agriculture, it means things like yield, quality, disease resistance, and so on. They're complex traits. There's a lot of genes contributing to variation in those traits. The question is how many genes, or how many mutations, to be more precise, and what's the effect of each of those mutations? Once we know a bit about our genetic architecture, what are the implications for genetic gain? What does it mean for how fast we can improve yields, improve disease resistance, and so on? So that's the first topic. The second topic is, given the architecture of the traits that we're looking at, how do we use genomic information that's increasingly available? So by this I mean SNP chips, genotyping by sequencing, and so on. How do we actually use that to drive faster genetic gains? And then for the people setting up genomic selection experiments, which I hope some of you are doing, we'll have a look at what drives the accuracy of genomic predictions, genomic selection. And finally, if we get to it, how do you handle genotype by environment interactions in genomic predictions? So we'll see how we go in covering that in an hour and 45. But first of all, let's look at this question. What is the genetic architecture of the traits we deal with, like yield, disease resistance, and so on? And what's the effect of that on genetic gain? So one of the wonderful things that happens is that we see genetic variation for almost all the traits that we look at. I can't really think of a trait where there's not a genetic component to the variation in that trait. Sometimes it's small, sometimes it's moderate, but the genetic variation seems to almost always be there for the traits that we look at in livestock and crops. And the question is it one gene or many? So here you can just see a picture of chickens. They're the same age actually, but very different weights. How many genes drive the differences between the weight of those chickens? And then for maize, different in cob size, how many genes drive those differences? Now this is where we're going to step back a bit to, in fact, 1918 and look at a model that was proposed to describe the variation in quantitative traits, complex traits. And it sounds really weird, this model. In fact, it's quite bizarre, but it is the foundation of modern animal breeding and plant breeding. And this model that all breeders use, sometimes without knowing it, is called the infinitesimal model. And what it does is it assumes that the genetic variation, so the differences we see between individuals due to the genetics, are the result of an infinite number of unlinked and additive loci, each with an infinitesimally small effect. So there's an infinite number of these mutations, and they all have infinitely small effect. I told you it sounded pretty weird. And this model is actually the foundation of animal breeding theory and plant breeding theory in some cases too. And it's been really spectacularly successful. But just to describe to you how you put it together, or how this model feeds into genetic gain, this is the little formula for genetic gain. And what it says is the amount of genetic gain you make is dependent on the intensity of selection. So that's how many lines you select out of the total number of lines to be parents, or the number of individuals you select out of the total number of individuals that you have information for. That's the intensity of selection. R is how accurately you do that. And by accuracy, we'll come back to this quite a bit, but this means how close do you get to the true breeding value, the true genetic value of the individuals when you're making these selection decisions. This is the genetic variation. I've called it sigma G there, but it's this thing. And this is where this assumption about the genetic variation being due to this infinite number of loci, all with an infinitely small effect, comes in. And then it's divided by the cycle time. So depending on how long it takes you to turn over a generation, might be 4 years, might be 12 years, you divide by that. So a very important point for later on. The longer the cycle time, the less genetic gain you make per year, of course. So that's our little formula for genetic gain. And as I said, some of these genetic gains have been spectacularly successful. So in chickens, if you can see the two chickens down the bottom there, over the last 30 years, I think it is, the time for market weight for meat chickens has decreased from 16 weeks to 5 weeks over those 30 years. So that's just a result of selecting out the chickens that hit heavier weights earlier in their life. So that's why chicken's so cheap in the shops, because we made such gains there. And that's a result of this genetic theory based on this infinitesimal model. Just in wheat breeding, this is just a cartoon of what the breeding cycle looks like. So you have your line development, field testing, and release. And this cycle turns out to be very long. It's up to, you guys can tell me, 8 to 12 years, depending on how fast you're taking things. But the cycle is very long. And part of that is because you can only evaluate some traits, like quality, right at the end of the breeding cycle. So in wheat, using much the same theory, gains have been steady at about 1% per year, or perhaps a bit less. And that's because this cycle time is very long. So the gains per year are only about those 1%. Okay, so that's the theory that we've been working with since about 1918, when Roald Fisher came up with it. But we know that that strange model of infinitely many mutations of small effect actually can't be true. And that's because we all have genomes. Our agricultural species have genomes. There's a finite amount of genetic material, maybe a very large amount of genetic material for a species like wheat, but it is finite. For humans and livestock, it's about 3 billion bases. And somewhere in there are the mutations that are really affecting our traits. And the real question is, how many of these mutations affecting the traits we're interested in are there, and what are the size of their effects? So for a typical trait, are there just a handful of mutations affecting the trait, in which case it looks very different from what we've been used to, the infinitesimal model, or are there many, many mutations affecting the trait? So let's have a look at some examples. One of the examples we looked at was proportion of black on cattle. These are dairy cattle, in fact dairy bulls, and you can see this one's got a lot of black on its coat, this one's got a lot of white. And you might think, as we did, that there'd just be maybe 2 or 3 genes that determined how black or how white the cattle's coat was. That's what we assumed before going into the experiment. And when we did a genome-wide association study, so we had about 600 bulls, all with their proportion of black recorded, genotype for 50,000 markers. We went in, we started dissecting the trait. We did find some genes of large effect. So this is one on In the little graph there, you see one on chromosome 6. So the x-axis there is the position on chromosome 6, and then you have the DNA marker and its significance on the y-axis. So this DNA marker is actually tracking a gene that has a big effect on coat colour. But by a big effect, it only actually explains 9% of the variation in coat colour. And that was the biggest gene we could find. And in fact, for this trait, it looks like there are hundreds of mutations determining how black or how white the coat is. So even for a simple trait like that, it's a complex trait. And then we get into even more complex traits, and a real classic is the percent of oil in the maize kernel. And there's this really beautiful experiment that's been going for 100 years or more now in Illinois, where they select for increased oil content in the kernel, and they also have lines that go down, so select for decreased oil content. And in this graph you can see the response that's been made. So this is generation on the y-axis, percent oil in the kernel, on the x-axis, percent oil in the kernel on the y-axis. You can see it just for the line where they're selecting for increased oil content, just keeps going up and up and up. When you select down, of course, you have a problem because you get pretty close to zero and you can't go beyond that. But you can see the dramatic increase. So what's driving that increase? Is it just a couple of genes or is it many? So these authors of this paper, they actually looked for mutations underlying those differences, and they found that many mutations were required to explain the difference. So they found 50 quantitative trait loci, that's regions of the genome containing a mutation affecting the trait. So they found that 50 mutations were required to explain just 50% of the variation. And they needed hundreds of others to explain the rest of the variation. So a trait like oil content in maize kernels is driven again by hundreds of loci, if not more. A few other examples. Yes?
I'm always curious about this Almai's experiment on the maize oil contents. So they say that it's due to the mutation, like we have the mutation, that's why we have the genetic gain, and that genetic gain wouldn't exhaust like for the next 50 or 60, 70 years. But my question is that might be the mutation frequency is very low compared to other effects. So how is possible, for example, in one breeding cycle, how much will be the breeding mutation frequency? It will be like very, very, very low, might be 10 to the minus 12 or minus 10. So how is it possible that this so low mutation frequency can give you the genetic gains for 100 years or so?
So what this is largely operating on is standing variation, mutations that occurred before the selection experiment. So mutations that have occurred since domestication or pre-domestication. So all those mutations that have occurred in the history of maize. And then you can imagine them segregating in the population. They start to select on oil content, and the frequency of those mutations starts to change, starts to increase a little bit. But because there's so many of them, the change in frequency per mutation is only small each generation. I can't remember if it was this experiment or others, but over 100 years, you do have a couple of extra mutations, just a few, maybe 5, something like that are new mutations that the selection picks up and starts to make progress. But it's very largely standing variation before the selection began. And it's the same in livestock and other species as well. Okay, another trait to look at is yield in rice. And there was this lovely paper came out in 2014, I think, where they actually had whole genome sequence data on many 517 lines of rice, and they started to dissect out the number of mutations affecting different traits, different agronomic traits, like yield. What they concluded is that their results implied many loci, multiple loci, with small effects contributing to the variance. Okay, human height. Human height's a really interesting one. Maybe if we all just stand up for a second, we can see the variation in human height. Okay, I think Kai actually wins, doesn't he? But it's not a competition, doesn't matter. But if you look around, there's a huge amount of variation in human height. And it's largely genetic as well. The heritability of human height is about 80%. 80% driven by genetic effects. Human geneticists are a bit, you can sit down there, are a bit obsessed by human height as a trait. They use it as a model complex trait because it gets measured in so many different experiments. And the surprising thing is when they start to dissect it out, look at how many mutations affect this trait, there's a very large number, and they all have really small effects. So the largest study to date is, this is 250,000 people measured for height with 1 million SNPs, 1 million DNA markers each, and then they go and dissect out, find the loci contributing to height. They find 600 of them, but the largest has about 1 millimetre of effect. That's the biggest effect. So it means between Kai, who might be the tallest person, and the shortest person, there's actually a lot of mutations that are different, all contributing to that variation, not just one effect. Okay, so just to conclude that section, for most complex traits, there's lots of mutations driving the variation that we see. It's not true for all traits. Some of the disease resistance traits, there are of course mutations with very large effects, so they do sometimes exist, but for most of the traits we look at, there's a lot of mutations of small effect. This has a, I guess, disappointing implication that the variance explained by individual markers will be small. So it means if you do a genome-wide association study trying to find the mutations affecting your trait, you may get lucky and find a big one, a big effect. Does happen. But for most traits, what you find are those mutations that you detect have a small effect. Just as a something I should point out, when you do a genome-wide association study, if you've ever done one, and you find a mutation, in your initial experiment, you might find that it explains 30% of the variance. You think, woohoo, I found a mutation with a big effect. The problem is, in that experiment, the size of the effect is always overestimated. Because of sampling error, because you set this threshold, you say it has to get above a certain significance level. The ones that do get over that significance level, they're overestimated by a positive error term. So they have their true effect plus a big error term. And so they're overestimated by that. So there's heaps of work showing that you discover your initial QTL, your initial mutation, then you test it in a validation population. And the size of the effect shrinks right back, sometimes by a quarter or something like that. So it's really important to validate your QTL in a new population. But anyway, that's just an aside. So just be aware when you're reading the literature, just ask, has the group publishing the experiment, have they validated the mutation in a new population? That's the only way to get an estimate of the real size of effect. So just be a bit careful with that. Okay, the thing we do have in our favour though is, I guess you could call it the genomic revolution. And as a result of sequencing the genomes of our species that we're interested in, we're covering a huge amount of information on variation at the genome level. And the most abundant form of variation we find are single nucleotide polymorphisms, or SNPs. And I'm sure you all know what SNPs are, but just to show you, between these two cows, if you read off their genome sequence at exactly the same point on the genome, that sequence will be identical, except where you have one of these SNPs. So this SNP carries it, this cow carries a G allele at this SNP, and this cow carries a C allele. So that's a SNP. It's just a base pair difference. There are other kinds of variation, like structural variants. They're important as well, but less common than the SNPs. Just to give you an example of how many of these SNPs there are, I'm just going to use humans as an example. The 1,000 Genomes Project, it sequenced, so read off the entire 3 billion bases of the genome sequence for 2,400 of us, humans, from 26 different populations around the world. And when they stacked up those genome sequences, so you can imagine stacking them all up on top of each other, they looked for SNPs. They found 88 million sites where there was a SNP. So there's a huge number of SNPs there. And in cattle, we find the same thing. We get about the same number, interestingly. In maize, I'm not sure what they're up to, but it's certainly in the tens of millions. And in wheat, I don't know. Does anybody know how many SNPs they've found in wheat? No? I suspect it's in the millions as well. Okay, the other part of the genomic revolution is being able to genotype individuals relatively cheaply. So sequencing the genome, it's still expensive. The cost is dropping all the time, but particularly for something like wheat that has a big genome, it's just too expensive to sequence multiple individuals at the moment. I think that'll change. But the other part of the genomic revolution is things like SNP chips, where you put, say, 50,000 of these SNPs on a SNP array. So this is a small glass slide that can assay multiple individuals for 50,000 SNPs at once. And there's SNP chips available for all the livestock species, plants for maize and wheat. I think there's a 90k SNP array for wheat, 9k, and there's others on the way as well. And the cost depends how good a negotiator you are, but it might be something like $50 or $60. That's either cheap or expensive, depending on where you're coming from. If you think 10 years ago that would have cost $500 to get that information, it looks cheap. If you're a breeder and you think about spending $50 per individual, it looks a bit expensive. But we can come back to that. Genotyping by sequencing is also being used very widely. This is where you sequence just a small part of the genome and get the SNPs in that small part of the genome. Okay, so that's our genomic information that we have available. The question is, and given our architecture of our complex traits, where we said there's lots of mutations affecting the traits we're interested in, how do we use this genomic information to maximise genetic gain? What are we actually going to do with it? If you go and genotype a wheat line with one of these SNP arrays, you might end up with 90,000 ACTs and Gs. That's not very, well, it's a lot of information, but not very useful. How are you actually going to use that in breeding? So that's the next topic. So the proportion of variation explained by each SNP is likely to be small. In other words, they'll be tracking a gene that has a small effect. That means for a lot of traits, not for all, there's some traits where you get a big boost from marker-assisted selection, but for a lot of traits, the gains are limited because of these small size of effects. An alternative is to use all the SNP information simultaneously. So if you have 90,000 SNP, use all 90,000. If you have 9,000 SNP, use all of them. The idea is you're going to use all that SNP information to track the entire genome. So all the chunks of genome that you have across your individuals and capture all your variants affecting the trait. And that's basically genomic selection. The difference between marker-assisted selection is you're not just tracking one part of the genome, you're tracking the whole genome simultaneously, trying to capture the effects of all those loci. So that is what genomic selection does. Okay, let's dive into genomic selection in a bit more detail. So the start of genomic selection, whether you're working in pigs, which this cartoon comes from, or wheat, or whatever species you're into, what you need to start is a reference population. You need, or it's also called a training set. You need a large number of individuals, and we'll see how large in a moment. A large number of individuals, so it might be lines, that have genotypes across the whole genome, plus trait records. And that's a really critical thing. You don't get anywhere with genomic selection unless you have really good trait records. So if you're trying to get genomic selection working for yield, you need good measurements of yield somewhere. For disease resistance, you need good measurements of disease resistance. Otherwise, it's just not going to work. So it starts with a reference population. And it might be, 2,000 individuals, genotype for 9,000 SNP, or 1,000 SNP, or something like that. Then what happens is, this is just a little cartoon of what happens. You're dividing the genome up according to the markers that you have. So this is a chromosome here, and M is a marker, a DNA marker, so a SNP, and the Q is a mutation affecting the trait. And it might have a moderate effect on the trait, small effect, or no effect at all. But you're dividing your genome up into all these different chunks, segments. Then the example here that I'm using is for growth rate of these pigs, but you can think about it for yield, for wheat, or whatever trait you're interested in. So what you do is you estimate the effect of each of those markers. So if it's 9,000 markers, you have to estimate 9,000 effects. So in this particular case, the marker has two alleles, A and T. If you have the AA genotype, so all the animals with the AA genotype, they have zero extra effect on growth. It's because that's where we're setting the baseline. If you get one copy of the T allele, you get an extra 200 grams of growth. In other words, it's saying that SNP is very close to a mutation that increases growth rate, and if you get the T allele, it increases it by 200 grams. If you get two copies of the T allele, two copies of the gene, improving growth rate, you get 400 grams extra growth. And what we do in genomic selection is we weight each SNP by the effect on the trait. So it's always the effect of the second allele. So in this case, the weight is 200, the effect of getting that T allele. So that's for one SNP. You just have to do that 9,000 times. And it's very good to do them all together as well, not just one SNP at a time, because this SNP over here, the first M, it might pick up the effect of that mutation as well. And unless you fit them all together, you can get double counting very easily, where you pick up the mutation with the effect of multiple SNPs. But we'll come to how you do that in a moment. Okay, so what you end up with is a weight for each SNP. And the weight is the effect of getting that second allele. So if it's A and T, the effect of getting a T allele. And that's what's called your prediction equation. It's just all those weights. So if you have 1,000 SNP, it's just 1,000 effects of getting the effect of the second allele across the genome. Once you have that, you're really in business. Because what it means is you can go into any line, even if it's a seedling, genotype it, and get a prediction for its genetic value for the trait you're interested in, just from the genotype. So you can come to your selection candidates. So you might have, I don't know how many, 20,000 seedlings that you want to know the genetic value for. You genotype them all, run them through the prediction equation, this produces your genomic breeding values. That's your prediction. And then you select out the best ones to breed from, to use as parents in the next generation. Okay, that looks simple in a cartoon like that. The trick with genomic selection is to get estimates of these SNP effects, the effects of the second allele. And the challenge here is the number of effects might be a lot bigger than the number of individuals you actually have. So you might want to estimate the effect of 50,000 SNPs from 2,000 lines or 2,000 individuals. If you tried to do that with least squares or whatever, so fit 50,000 effects with 2,000 observations, you just can't do it. The system is over-parameterized. This is called the P greater than N problem. So the number of parameters, which is the number of SNPs, is a lot greater than the number of lines. So you need methods that can deal with this. How do you actually deal with this problem? And this is the crux of the methodology for genomic selection, is getting estimates for many times more SNP effects than you have observations for. Okay, just a little bit of statistics for you. Bit hard see how we go first thing in the morning. But just to say what we're actually doing here is fitting a linear model, a linear mix model. So what we do is, this is to get the prediction equation, to get the effects of the SNPs. We say that the phenotypes we observe, so this might be yield. You have yield for estimates of yield for each line. That's your phenotypes, is equal to the population mean, so just the average of the population. So we're expressing everything relative to the mean. So this is the mean here. And it just says all individuals get a dose of that mean, plus the effect of all the SNPs summed together. That's what this XJ thing does. The X tells the equations what genotypes the individuals actually have. So do they get a AA? Do they get an AT? And so on. Okay, if you're just doing least squares, or if you're just doing one SNP at once, that would pretty much be the end of the story. You'd fit that little model, solve for the SNP effect, and job done. The problem is when we have 9,000 SNP or 50,000 SNP, we can't do that. We've got more parameters than effects. So how do we deal with that? And what we do is we use this BLUT methodology, best linear, unbiased prediction. And what it says is all our SNP effects are very small. They come from a normal distribution, which I've got here. So this is our SNP effects. This is the size of the effect on the x-axis, and on the y-axis is the proportion of those SNPs with that size of effect. So what it says, like all normal distributions, it says that many of the effects are close to zero. The chance of getting a large effect is basically zero, but you have some that are a bit bigger. But many have a small effect. So by using this information, it actually allows us to solve for the SNP effects. This is, if you ever hear Bayesian statistics talked about, this is in fact a Bayesian approach where your prior assumption, you're making the assumption before you fit the model that your SNP effects really are small. There's very many of them. They all have a small, close to zero, but not a zero effect. And this is what lets you, by borrowing this information in, by pulling this information in, this is what lets you actually estimate 50,000 SNP effects from just 2,000 records. It's because you borrowed in information from somewhere else. And I'll just skip that. Now, you don't have to remember this at all, but this is just the system of equations where you solve for these SNP effects and you pull in this information about the SNP effects being normally distributed. So if you were, just for the people who do solve these things by least squares and so on, what you get out of the equations is the effect of the mean and the SNP effects. If you were fitting it as least squares, it would just be something like this. If you were fitting it as a random effect, pulling in this information that it's normally distributed and the effect of the SNPs is small, you have this additional bit in the equations. But you don't have to remember that. How many, just for my benefit, how many people have played with R before? It's a few of you. What I might do is just go through a small R example of this to show you solving for these SNP effects and how the equations actually work. So, and I'm very happy to give you this little bit of R code that I'm about to go through and the data sets as well. So Lee, my email's on the sheet, isn't it? So if you want the R code I'm about to show you, please just email me and I'll give it to you. But it's also implemented in R packages like RRBLUP, so you can do it there. But I might just go through this small example so you can see it in action. But I will have to sit down so I can manage it. First of all, the data set that we're going to use. This is the genotypes coded up. So it's exactly like we did here to get the X matrix, except there's 10 SNPs now. So my X matrix has 10 columns going across. So it's just one line per individual. In this case, it's a dairy cattle data set. So these are dairy bulls. This first line, this first one, says this bull has the genotype. What would it be going on that? AA? That's right. Yep, AA for the first SNP. If the second SNP was an AT SNP, it's also AA, and so on. There's 10 SNPs going across there, and there's 325 bulls. with genotypes. So there's no ID there unfortunately, that would make it a bit easier, but it's just 325 bulls going down. So that's our X matrix. So that's recoding, say, 10 SNP genotypes we get back from the lab. The other bit of information is a trait record, so the phenotypes. Okay, and these are the phenotypes. So this is actually protein percent in milk, is the trait here in this dairy cattle example. So the first bull, he has an extra 0.1%. Actually, it's in his progeny's milk, his daughter's milk. This bull, his daughters have an extra 1.2% protein in their milk. So quite a lot extra protein. And there's, again, 325 lines going down there. I'm just showing you the first few lines. So what we're going to do next is, in our little bit of R code, we're going to take that data, the genotypes and the phenotypes, and get the estimates for the SNP effects using this BLUP methodology. So when you hear BLUP, what you should think is we're pulling in information that we think the SNP effects are normally distributed, come from a normal distribution, they all have a very small effect. That's the information we're pulling in. So just to show you, it's actually a bit small. Okay, I'm just going to make that font size a bit bigger. Okay, so those familiar with R may or may not be happy with this. I can give you this script, so you certainly don't have to remember it. But just to tell you what it's doing, it's reading in that X matrix, that matrix of SNP genotypes. It's reading in the Y, the vector of phenotypes, the trait records, and then it's solving for the SNP effects. So it's solving the system of equations for the SNP effects. And all it does is build up the system of equations. So build up the system of equations required to solve for those SNP effects. So just to show you what you get out of it when you run that. Okay, so remember we had, to change that. Remember we had 10 markers. So what we should see when we solve this system of equations is we should see a solution for both the mean effect in the population, and then 10 marker effects. So let's see if we get that. Do you know how to increase this font size? I can just put it into Notepad, might be easiest. Okay, so this is the solutions. The first one there is the mean effect of the population, so that's minus 0.133. The rest are the effects of the SNPs. So the 2 there is actually the first SNP going down to the 10th SNP here. And each of those is the effect of the second allele. So if T is the second allele, it's the effect of getting that T allele. So which SNP has the biggest effect? Yep, it's actually SNP 6. It's in the solutions vector, it's element 7, but the first is the mean, and this is number 7. That's actually quite pleasing because in this dairy data set, I knew there was a QTL, a real QTL for this trait close to SNP 6. So it's actually found that. Now this is the effect of the SNPs, the effect of the T allele. We've solved with our BLUT method all of those effects together. Just to show you something, when you read the literature on genomic selection, you often hear about shrinkage. Okay, if you read about, pick up any of the BLUT papers, you read about how much the SNP effects are being shrunk. And they do get shrunk, they get compressed back to the mean, because we make this prior assumption that the SNP effects are normally distributed and have a small effect. They're shrunk because we assume they do have a small effect. But just to show you that, if I rerun this, assuming that normal distribution is narrower, so has a smaller variance, so the SNP effects are even smaller, what I expect to see is the SNPs get shrunk back even further. So let's just test that. Let's rerun this, but saying the SNPs come from a normal distribution with even smaller variance. So applying more shrinkage. To do that, I just change this parameter lambda. What the lambda does is tell the equations the relative size of the environmental variance, the noise, relative to that genetic variance. So a bigger number means there's more noise relative to the size of the SNP effect. So let's just rerun that. But now we're making the assumption that the SNP, the normal distribution, has 10 times less variance than it did before. So our lambda goes up by a factor 10. Okay, these are our new effects. Where now I'm saying to the, and this is under your control when you run these BLUT methods. I'm saying the variance is even smaller, so the SNP effects are smaller by changing that lambda parameter. Now if you look at SNP 6, which was the biggest one, we've shrunk it right back. The effect previously, if I just go up to the previous solutions, it was 0.26. It's now been shrunk back to 0.1. Okay, so when you run these BLUT models, you can actually control how much you shrink the SNP effects or not. So you can do that in a couple of different ways. One is you can just play with how much the shrinkage is by changing the lambda. The other is to use something like cross-validation to get what is a good amount of shrinkage for the SNP effects. Yep.
So with lambda, what's the kind of variable, like thresholds for us to set? Like, is there from to set, or is it just trying lots and lots of numbers to see which one is the best, and how would we know that?
Okay, so you can't use 0 because that just makes it into a least squares model, and that's not going to work. So you have to use something bigger than 0, and it really depends on how many SNPs you have. if you have 50,000 SNPs instead of 10, what you want to do is say the effects of the SNPs are all really small because we have 50,000 of them. So you want a big lambda, a really big lambda. And in practice, it's often something like 1,000. But I'll show you how you can get a good estimate for lambda from cross-validation. So this is the way cross-validation works. Imagine you've done an experiment and you've got 1,000 lines with yield phenotypes and they're all genotyped for, say, 1,000 SNPs. Okay, so here's the whole data set with 1,000 lines in it. What we do in cross-validation is we drop out, say, 20%. So you drop out, what's that, 200 lines out of your data set, and you keep them completely aside. So for the moment, you don't use their information at all. So you've got 800 left here, and you try a lambda value. You just guess what lambda might be. So we say it's 10. We run our BLUT model just like we have done here, and then once we've done that, we get our effect of our SNPs. We get the SNP genotypes for the individuals in the group we've left out, and that's just their genomic breeding value is their X matrix, so their 0, 1s and 2s, times the effect of the SNPs. Okay, so if we've got 10 SNPs, our X matrix will have 10 columns, and we'll have 10 effects here. And it's exactly what's up on the thing there. So this G is just the effect of the 2 allele at each SNP. Then we can assess how well we've done, how well this lambda value works, as the correlation between the GBV and the phenotype for those lines. So these 200 lines, this 20%, they each have a phenotype for yield, say. We just correlate their genomic breeding values that we get from their SNPs, their SNP effects, and their phenotypes. And that might be 0.3. Let me do this differently. So when we use a lambda of 10, we got 0.3. Okay, so that's one estimate. Then we go back and we try the whole thing again with lambda equals 100, for example. And say now we get 0.4. So in this case, a lambda of 100 is better than a lambda of 10 because it's giving us a higher correlation between the genomic breeding values and the real phenotypes for those lines.
So it means when we have the high correlation, the prediction accuracies are more higher?
Yes, yes. This isn't exactly the prediction accuracy. You have to divide by how heritable these phenotypes are to get the accuracy, to the square root of that, but it's very related to the prediction accuracy. Yep. So what you can do then is draw yourself a graph of lambda and accuracy, or the correlation between the GBV and the Y. And you try different values of lambda your line might go up like this and then come down. So your best value of lambda is here. And that's how you get the best value of lambda for your data set. That's actually a one-fold cross-validation, because we've dropped out 20% of the data. Then we'd go back and drop out another 20%. And if you hear five-fold cross-validation, it means you've cut your original data set into 5 different chunks and done this 5 times. And then your best lambda is the average across the 5. Okay, so you just step through different values of lambda until you have the highest accuracy. And that actually works really well, that cross-validation. We do that routinely for genomic evaluations. Okay, so I'm going to shift. Yep.
Between those two data sets, if you look at the values for the first individual, in the first case it was negative and it turns out to be positive in the second case.
For the first SNP.
Yeah.
Yep, that's a good point. Okay.
Is it because the number is quite small?
Yes. This is just noise. Those don't actually flip.
Yeah.
But because the noise, the error variance, is much bigger than the SNP variance, what you see are the SNP effects are tiny, and when you shrink them right back, you're basically back to no real effect. It's just noise. So it's only a few of them are above the level of noise. Yep. But the great thing is you have shrunk them back. So they are very close to 0, so they don't bugger things up. If you didn't shrink them back, and they still had a moderate effect, but it was the wrong direction or something, that would cause your predictions to be terrible. And that's actually what happens with least squares, where you don't shrink things back. Okay, so as I said, please email me if you want that little script for doing genomic predictions. It's also in the RR-BLUT package. In fact, the RR-BLUT package is much nicer because someone else wrote it. But I can give you these little data sets as well. Okay, we're going to have a break for 5 minutes in a moment, but just to take you through one more thing before we do that. What we just went through was taking our data set and getting the effect of each SNP, getting a prediction equation. Once you have that, it's great, because any individual with a genotype, you can get a genomic prediction just by multiplying the vector of genotypes that it has, number of copies of 2 allele at each SNP, by the effect of those SNPs. Once you have your genomic predictions, your genomic breeding values, you can start to select your individuals to breed from. If you read through the genomic selection literature, you may have heard of another way of doing these genomic predictions. And this is GBLUP. This is basically genomic relationship BLUP. And what I want to just convey to you is this is an exactly equivalent model, but what you do in this model is you first build the genomic relationship between all the lines and use that to help your genomic predictions. So instead of solving SNP by SNP, in this GBLUP model, you first build the genomic relationships between the lines. So the genomic relationship is what proportion of the genome do a pair of individuals actually share in common. Okay, so just to jump out a bit, if you have a full brother or sister, what proportion of the genome are you expected to share with them? Of your DNA, with your full brother and sister, what proportion of your genome do you share? Are you expected to share? 50%? Correct. Yep. So with your full brother or sister, you share 50% of your genetic material. But in reality, because your mother and father have chromosomes and they're diploid, so they have the chromosomes they got from their mother and father, and they can pass on either one of those chromosomes, you can share actually anywhere between 0 and 100% of your genome with your full brother and sister. In reality, it's more like between 30 and 70% because there's so many chromosomes. And if you think about your full brothers and sisters, you might think that, yeah, some of them are more similar to you than others. And this GBLUP, it makes use of that information directly. Okay, so it's an alternative way of doing the genomic BLUP. So it first estimates what proportion of genome two lines share between each other. And if it was a human example for full brothers and sisters, that might be anywhere between 30 and 70%. And then it uses that to solve for the breeding value. So if you want to think about it like this, in your reference population, you might have a whole bunch of lines, and then you come to evaluate another individual. And with this genomic BLUP, what you do, you calculate the genomic similarity or proportion of genome with all those individuals in the reference, and you find one that's really similar, has a high proportion of the genome that it shares. And what the equations will do is say, hey, I've seen this line before, I've seen this DNA before, to be more precise. I'm going to give this new individual a phenotype, a trait record, or an estimate of the trait that is pretty similar to this individual where I've seen this DNA before. That's what it essentially does. So just to go through that again, if you're evaluating a new individual, you calculate the proportion of the genome in common with all the reference individuals. If there's a really similar reference individual, high proportion of genome shared, you're going to borrow that individual's trait information and say, hey, I've seen how this DNA performs in the field before. I'm going to give a similar trait value to this new individual. And this is the way GBLUP basically works. But, yep.
So does this GBLUP consider all the possible combinations for the identity by descent? So what does that mean? If we have 100 individuals, will it calculate the identity by descent 1 by 2, 1 by 3, up to 1 by 100, and 2 by 1, 2 by 3 up to 100? So does it consider like all the possible combinations for IBD?
It considers the IBD between every possible pair of individuals. Yep. So if there's 100 individuals, it considers the possible IBD between all those individuals. It's actually not quite IBD, it does it by identical by state, but you have so many SNPs, it's very close to being IBD, as an aside. Oh, I should just write up here. One thing I left out here was to get the genomic relationship matrix, which we call G. It's just X times the transpose of X. So if you remember, the X was the 0, 1s and 2s. It's the 0, 1s and 2s times itself. But flip through 90 degrees is the transpose, divided by the number of markers, essentially. So if you get your genotypes back from the lab, and you want to calculate the genomic similarity between your individuals, you can do it just by constructing this X matrix, multiplying it by itself, and dividing by the number of markers. And I would suggest that's an extremely useful thing to do, and I'll show you why. Okay, so this is the relationship matrix, the genomic relationship matrix that we got from, I think it's 9,000 SNPs, but you get a very similar picture from 1,000 SNPs amongst a worldwide collection of wheat accessions. This is the Watkins collection, if you've ever heard of that. And so what this thing is, it's actually a matrix, the genomic relationship matrix. Each little dot is the genomic relationship between a pair of individuals. And I think there's about 300 individuals across there, so it's 300 by 300. You can see on the diagonal here, the individuals share 100% of their own genome, which is what you'd hope. Something's gone seriously wrong if they don't. So this is the individuals with themselves, but the other dots are what proportion of genome each pair of individuals share. And it's basically a white is a 0, so share no genomic relationship, and red is 100%. So this is a very good thing to do. You can use it in your genomic blob, but it's a very good thing for quality control as well. Because what you see is a bit of structure in here. So, for example, amongst the Australian wheats, up here, there's a block that is all quite closely related to each other. They have a high degree of genomic relationship. And that's because they have similar parents in common, similar ancestors. If you go back, there's a few lines that were the founders of that population. And there's other blocks of structure too, for example, amongst the Chinese spring wheats as well. And here, what's this one? This is Iranian wheats. And here we have a group of individuals that's extremely closely related. So it's a good check of your data just to see you're getting back what you'd expect. If you get back no relationship amongst the lines, that means there's a problem. If you get back two lines that have 100% relationship, it means you've essentially genotyped the same thing twice. And they might actually be different names, but 100% relationship, the same thing. Or it could be a sample mix-up as well, where you've mistakenly genotyped one sample. So it's a good thing to try and put together. Yep.
Maybe a very stupid question, but the ones which are white, so no relationship at all. Isn't that strange? All of it is wheat. So there must be some relationship.
But it's a good question. You're exactly right. And all wheat that we breed from has some relationship, but it's expressed relative to a base of 0. So the least related lines are put to 0. That's why you get the whites. If you didn't do that, it would all be some degree of yellow. That's right. Yep, spot on.
Yeah. Is it same as association genetics, where you take into account the relative kinship between the individuals you are working on?
It's exactly the same. Yep. It's the kinship estimated from the markers. That's what it is. Yep. And in this case, instead of correcting for population structure, like you do in a GWAS, we're actually using this to help us predict. And just to go through it again, say you were trying to predict this line just here. what it says is all these other lines in this block, they would do a good job for you. So the GBLUP would predict that this line here has similar genetic value to whatever phenotypes you observe for these lines. So if you get good yields for these lines, it'll predict a good value for yield for that line because they share a lot of their genome.
Sorry for this question. Your estimate would also depend on the number of markers used for genotyping. Like few of the markers might give like less information.
Yep.
Like here in this case, you have say about 300 markers and 300 individuals, right?
Yep. This is 9,000 markers. If you have less than that, there's more error in these estimates of relationship. And if you have like 300 markers, you can estimate the relationship between full SIBs still very well, but more distant lines becomes very more difficult.
So that would affect your output.
Yes. Yep. Yep. The GBLUP actually borrows to predict, say this last line here, it actually borrows information from all these other lines, depending on and in relationship to how related they are. Okay, so we might just take a quick 5-minute break. So you can just stand up and walk around. But please be back in 5 minutes. Okay, so we'll get started again. So we've been through the architecture of complex traits, saying that often for a lot of traits there's many mutations affecting that trait. We've been through different methods for genomic selection, how to actually use your SNP data. The next topic is how accurate will your genomic predictions, your genomic breeding values be with a certain size of reference population and other parameters. In other words, to phrase that differently, how big do you have to make your reference populations to get a certain accuracy and to have a noticeable effect on the rate of genetic gain? So just to remind you, put this down the bottom of the slide, but this is our little formula for genetic gain, and it says that the amount of gain we're going to make is a function of how accurate our breeding values are, in this case our genomic breeding values, divided by the cycle time. So really we want them to be as accurate as possible. Just to define accuracy, because it's pretty critical here, I'll just show you what two different accuracies look like. So in this first graph, we have an accuracy of 0.6. What does that actually mean? What does an accuracy of 0.6 give you, or what does it mean? So what it means is how well we can predict the true genetic value of an individual. So here, this is a Rust example actually. We have on the x-axis a predicted Rust score. So this might be a genomic breeding value for a Rust score. And on the y-axis is a real Rust score. So this might be from evaluating a line many, many times, getting a very accurate breeding value for that. An accuracy of 0.6, although it sounds impressive, it means that, there's an example here, an individual with a good prediction for Rust score, or in this case very susceptible to Rust, 5, its real Rust score might not actually be that good. On average across the lines, there's a reasonable agreement between the prediction and the true Rust scores, but it's certainly not perfect. Amongst this top group here, that you might select on genomic breeding value, their actual Rust scores can range anywhere between 2 and 5. So there's a big variation there. So an accuracy of 0.6, it says on average you're making the right decision, but for any individual line it can actually move quite a bit when you get its true or a very accurate breeding value. You get a higher accuracy. If we have an accuracy more like 0.8, it means it's easier to pick those individuals with the genetics that we really want, basically. So if we predict out the 5s, then the group that we'd pick, the top, would be their true value. So they all have about 5 for genomic breeding value. Their true value has a much tighter range between 4 and 5. So the accuracy really tells you what you can do with these genomic breeding values. And this is a bit crude, but I'll put it this way. With an accuracy of 0.8, if you're getting that good an accuracy, you start to be in the position of picking winners. So you can say, this individual, based on its genotype, is just going to perform really well. So it has a high true genetic value. If you get an accuracy of 0.6, that's still reasonable. If it's 0.3, you've got a much more cloudy picture. It means if you pick one individual, you could be very disappointed, because when you come to a field trial, its actual value could move around quite a bit. So with an accuracy of 0.3, you might be in the position of being able to cut off the really bad individuals, get rid of them, but you're not really in a position to pick winners. Once you get up to 0.8, which is where dairy cattle are actually, or a bit higher even, then you can start to pick winners. So just be aware of that use of genomic breeding values. Okay, so we'd like to be at 0.8, would be ideal. How do we get there? So that depends on the size of your reference population and the heritability of the trait. So this is factors affecting the genomic prediction accuracy. So the factors that do affect it are the size of the reference population, how big it is, the bigger the better, the heritability of the trait. For traits that are more heritable, your accuracy is going to be more higher for the same number of individuals. So those two things we know. That's determined by how much funding we have, how many genotypes we can do. The heritability, you can play around with that depending on your trial design and so on. There's also the underlying heritability of the trait. The thing we don't know is this number of independent chromosome segments. So that's how many chunks of DNA are floating around in the population that are different, that are actually different chunks of DNA. And it goes together in this little, sorry to hit you with more formulas, but this little formula down here. So the accuracy is square root of number of individuals times the heritability divided by the same thing plus this number of chromosome segments. So that's what we don't know. Let's work out how to calculate that. So this is also called the effective number of loci for a trait. And it's basically dependent on your effective population size, the genetic diversity of your population. The bigger your diversity, the more chunks of different DNA you have floating around in the population. You can imagine if you've got very diverse lines, you can have lots of differences in the DNA for a given point in the genome for those lines. And just to show you where that comes from, it actually comes from the number of ancestors used to found that population. So if you have a breeding population and you only have a few lines that were the founder of that breeding population, then the number of different chunks of DNA floating around is going to be pretty small. And I'll just give the example here of dairy cattle, Holstein bulls, black and white dairy cattle. The number of new bulls used in the global population of dairy cattle each year is about 10. The number of different bulls used each year is about 10. So the genetic base, if you were a conservation geneticist, you would think this population is on the edge of extinction because the genetic diversity is low. It's not true because there are millions of dairy cattle, but because there's so few founders, the genetic diversity is narrow, let's say. So these are these key ancestors, and this is the lines or the individuals used to found your breeding population. This is their different chromosomes that they carried, their genomes. And when you make crosses between those founders, and you do that for multiple generations, the current generation, these are the current generation of dairy cows, but you can think of your current breeding population, it's just a mix of these ancestors' genomes, all chopped up due to recombination. And the chromosome segments, if we take this bit of the genome, here there's the blue one, there's a purple one, a blue one and a green one. There's about 4 different bits of DNA there that come down from the ancestors. And it turns out you can calculate the number of different bits of DNA, this independent chromosome segments, as 2 times the effective population size. That's the genetic diversity. It's actually the number of new breeding individuals that are used each generation, times the length of the genome. Okay, that's how you get your number of independent chromosome segments. So for wheat, I can tell you the effective population size is about 50. It's actually even less than dairy cattle. Dairy cattle is about 100. Wheat, this is for breeding populations. This isn't for diverse germplasm collections and so on. It's about 50. And then you multiply that by your genome size to get these number of chromosome segments. So what's the genome size in wheat again?
17.
Sorry?
17.
Okay.
17.
17. 16,
Yeah.
17. Yep, that was the number I had in my head. Yep. Okay, once you have that, there's actually the size in Morgans, so genetic distance. So it'll be that times 100, I think, something in that order. Once you have that number, so you calculate effective population size is about 50, you times that by the length of the genome, but in Morgans, not in base pairs, which will be per megabase is about 1 Morgan, I think. Let me do this little calculation. So you need to do your L in Morgans. People remember learning about Morgans in high school or university? Yep. So this is genetic distance, and it's basically the number of crossovers. But very, very crudely, 1 megabase, so that's 1 million base pairs, equals 1/100th of a Morgan, 1 centimorgan. So if you have 100 centimorgans, equals 1 Morgan. That's extremely crude, and as you know, there are bits of the genome that do recombine and bits that don't, but it's a very rough guide. So in dairy cattle, the genome's 3 billion bases, so it's about 30 Morgans long when you calculate it. You can work out what it is for wheat, approximately. So this formula is potentially quite powerful because it tells you how big you need to make your reference populations to get a good accuracy of genomic prediction. First thing we need to do is just convince ourselves that it does really work, this formula. So just Quickly, a little dairy cattle example. We had different sizes of reference population ranging from 1,000 up to 6,700. The heritability of our trait is 0.9, which sounds spectacularly high until you realise these are all dairy bulls and they have 100 offspring each, and those 100 offspring are evaluated. So it's a progeny tested population. So it's very accurate phenotypes. They're almost not quite perfect predictor of the true genetic value, but pretty close. And as I said, the effective population size here is 100. Okay, so just to show you what the graph looks like, here's the number of bulls in the reference. So this is the size of the reference population from 0 up to 7,000. The y-axis is the accuracy of genomic breeding value for a selection candidate, for a young or an individual without a phenotype of its own. This dotted line here is from the little formula. So that's just this formula. So we're increasing the number in the reference, this NP. Heritability stays the same. So that's the prediction, and this is what was actually observed when we had, say, 2,500 individuals in the reference population. So the green line is the accuracy we expect from the little formula, and the blue dots and triangles are what was actually observed in this experiment for the accuracy of GEBV given a number of individuals in the reference. So it tells you it works pretty well. The observed, which is the dots and the triangles, lines up with the prediction pretty well. So that's just to try and convince you that this little formula works. Okay, if you accept that is true, that works, what does it mean for the size of our reference populations we need? Basically means they need to be really big to get accurate predictions, accurate genomic breeding values. So along the x-axis here, we have the size of the reference from 0 to 20,000. On the y-axis, we have the accuracy for individuals that only have the DNA markers. They don't have a phenotype. If you have a high heritability, like 0.8, you do pretty well. Even with, say, 2,000 individuals, you're getting accuracies of 0.65, 0.7. So that's good. If you have a low heritability, I'm sorry, but the news is not that great. You just need very large numbers of individuals to get genomic selection to give you accurate breeding values, accurate predictions of genetic merit. So for low heritability traits, it works, but you need large numbers of individuals in the reference. And we've observed that in practice as well. For traits with heritabilities of 0.1, you just need large numbers of individuals. In crops, you're a little bit lucky, I think, that you can play around with the heritability by doing field trials, designing really good field trials. You can actually increase the heritability for a line by designing better and better field trials, more and more plots and so on. So that shifts you up these lines. But still the message is the same. You need a large number of individuals. Okay, so reference populations for genomic selection. Just a little bit on the design of these. We need a large number of individuals to make it work. You can also get a bit of a boost if you have individuals in there that are very close to the candidates you're trying to predict. So say a really good thing to do is if you're trying to predict some progeny, if you have the parents in your reference population, because then the relationship is really close. Effectively, you only have a small number of chromosome segments, big chunks of chromosome that come down, and they're easy to estimate. So if you know which individuals you're trying to predict, you can get a bit of a kick in the accuracy by having closely related individuals in there. So if you're trying to predict a breeding population or offspring from a breeding population, it's really good to have the parents in there, because that'll give you a boost in the accuracy, better than what these numbers would suggest. I'm just going to skip that bit. So that's a key message, is try and get into your reference individuals that are close to what you're predicting. But be a bit careful here, because if they're all really close to the individuals you're trying to predict, you might be able to predict those individuals, but you can't predict anything else because you haven't seen their DNA before. And the classic with this is when you do a biparental cross, you've got the progeny from that. You can predict those really well. So you can predict any progeny from crossing the same two parents again pretty well, but you can't predict anything else. There's quite a few examples of that in the literature. So your reference, your training set, really has to be a balance between individuals that are close to those you're trying to predict, and to get enough diversity in there so you can predict all the lines that you might come across. If you haven't seen the DNA before, basically, you're not going to be able to predict it. So designing these reference populations takes a bit of thought. You have to make them big to get accurate predictions. That's because the effect of the genes are all pretty small. But you want to have individuals that are close to those that you're trying to predict as well. And getting that balance right takes a bit of thought. Okay, just to show you an example, just check the time here. Yep. So just to show you an example of genomic prediction, and I have to say I'm definitely no expert in rusts, but here, this is some work we did a little while ago where we did do genomic prediction for resistance to rusts. So we looked at, in this little example, just to show you how it works, we looked at 3 diseases: leaf rust, stem rust, and stripe rust, and the phenotypes were these scores from 1 to 5. I can't actually remember which way it goes. Which way does it usually go?
The lower the better.
The lower the better. The lower is more resistant. Yep. So I think I had that the wrong way around before. Sorry for that. I told you rust was definitely not my area of expertise. Okay, so the reference population here was 2,069 lines, phenotyped in 4 different locations in Australia, and genotyped with 90,000 SNPs. So, and this was done using GBLUP actually. So if you can imagine what we did, we worked out the genomic relationship between all those lines from the 90k SNPs, and we fed that genomic relationship into our GBLUP to get genomic breeding values for our lines. And to get an estimate of the accuracy, we used cross-validation, like I talked about before. So we dropped out, say, 200 lines out of that 2,069, estimated genomic breeding values for them without using their phenotypes, and then correlated it with their actual rust scores. Okay, and this is the accuracy we get in the cross-validations. So for leaf rust, it was 0.51, stem rust 0.62, and stripe rust 0.56. So I was actually quite happy with those accuracies. I thought they were reasonably good. So just to show you the picture, this is an accuracy of about 0.6. So if you wanted to select out the most resistant lines, here of course down this end, not up this end, you've still got quite a range. If you select out the most resistant on the genomic breeding value, you've still got quite a range in what the true scores could be, but at least they're reasonably correlated there. It's certainly enough to get rid of the poor lines out of the experiment, that's for sure. Now, a point for discussion is, as you know much better than I do, there are already a host of genes and mutations that affect rust. When we look at our genomic predictions, we can see that the 90,000 SNP pick up those mutations pretty well. So they're actually included to some extent in these predictions. You may be able to do better if you, well, in some cases you can do better if you genotype those mutations directly and add that in to the prediction. What we do find though is that it's not just those known mutations, there's a host of smaller ones as well that seem to be important. How am I going? Got 5 more minutes? Okay. Okay, just to finish up then, just a tiny little bit on G by E. One thing that I've learnt moving from animals to plants is genotype by environment interaction is much more important in plants than it is in animals. We don't see much of it in animals. We see a lot of it, there is a lot of it in plants. And basically, well, you can define it in multiple different ways, but one way of saying it is, or expressing genotype by environment interaction, is some genes are more important in some environments than others. So for yield, for example, if you are in an environment where you have rust coming through, those rust genes or protective genes are very important because they impact on yield, whereas in another environment where you don't have certain kind of rust, they're not so important. So that gives you genotype by environment interaction. It's just one way of expressing it. And just to show you this, to unpack it a little bit more, so the experiment for wheat rust I just went through, it was, the rusts were actually measured in, well there it's got 3 different locations, and there's a map just of where these sites were. So here, 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 and right over in Western Australia as well. Oh, there's one here as well. So the question was there genotype by environment interaction between those sites for the different rusts? And what we've started doing is extending this GBLUP methodology to take into account multiple traits. So what we're doing is treating the resistance, disease resistance, as a different trait in each environment, to take account of the fact that some genes might be more important in some environments than in others. So unfortunately we don't have time to go into this, but I'll give you a paper where you can read about it. This multiple trait approach is one way of dealing with G by E, and it's probably the simplest as well. And with GBLUP, it becomes quite powerful. So just again, you treat, for each environment that you have, you treat the phenotype as a different but potentially correlated trait. And that allows for the fact that some of these genes, some of these mutations, are more important in some environments than others. And just very briefly to show you, we can get good genomic predictions in some of these environments. So here's the 3 different environments, but the accuracy is quite low in other environments. And that may be because we haven't captured all the genes important for the trait in that environment. So for leaf rust, we do pretty well on the east coast of Australia. Horsham, it's high. Cobbitty, it's high. But in Western Australia, it's quite low. And that's just saying we're not picking up all the genes affecting rust resistance in that other environment, which is quite different. And the same is a little bit true, though that's the most extreme example that we have, is the leaf rust. Okay, just to finish up then for the whole morning, take-home points. The variation that we see in many complex traits is the result of these mutations of small effect. So many of the traits we look at have many mutations that explain the variation in those traits. So what does that mean? It means we need markers across the whole genome to pick up all those genes affecting those traits. And genomic selection makes use of this. It estimates the effect of the SNPs across the whole genome, not just one SNP. And you use the SNP information to give you these genomic breeding values, which are basically the sum of the SNP effects across the whole genome. So you capture all the variants. A really important point to emphasise is that once you have this prediction equation, it's really good for your breeding program because you can start to select out the best individuals early and start to breed from them. And if you can do that, you can cut down the cycle time, in some cases quite dramatically. So you might go from an 8-year or a 12-year cycle time down to just a couple of years. And our little formula for genetic gain tells us that's because it's a function of, or you have to divide by the cycle length, that's going to really speed up the genetic gain. So if you have the opportunity to breed from individuals early, then genomic selection can really speed up your rates of gain. Of course, that depends on the accuracy that you're getting. If the accuracies are really low, the way you use genomic selection might be to get rid of the poor lines early and not take the cost of phenotyping those. This G by E, genotype by environment, it's definitely there. One way of dealing with it is this multi-trait genomic blot, where you treat the trait in the different environments as a different trait and get the correlation between them. And that seems to work reasonably well. Simple, but seems to work. Okay, just acknowledgements. The group at University of Queensland, but I'd also like to acknowledge the group I used to work with and still do some work with down in Victoria at the Department of Ag there. And there's a series of papers that I've listed here that describe those genomic predictions for rust. I think they're all in TAG, aren't they? Yep. And there's also this paper where we tried that multi-trait genomic prediction approach. So you can have a look at that. And some of that rust resistance work was funded by Dow AgriSciences as well, which was great. Okay, I'll leave it there.